Okay. Hey, everybody. And this is the next episode of Podlodger Podcast. And this is the new season is starting. And then this is going to be a special episode. And we have a guest, and who was my the TA for my Neuroscience of Simple Learning course, and who is currently working in a UBC Dr. Rankin's lab. And hi, Nick.
Hi, Chiro.
Yeah, it's very nice to see you. And it's been a while since the last time we met, right?
Yep, yep. I'm happy to be on the podcast.
Thank you for accepting the invitation and coming and joining me on the podcast. I
No
appreciate it.
problem.
Okay, so I think let's start with your. I think I very briefly introduced you as I know. In the class, right? We talked a lot about the class stuff, and then we never talked about your personal stuff, and then how you get to this position, and then previews how your the career path to get to this position. Could you
Yeah.
please introduce to our listeners?
Yeah, so I'm a PhD student at UBC in
Mm-hmm.
Dr. Rankin's lab. And I guess, do you want my career path
Yeah,
briefly?
and then starting from how was your undergrad and probably where you grew up and how was your high school or like the and then undergrad and what was your master's and how do you get chose this PhD and then love this academia.
Okay. Absolutely. Well, high school was I went to Point Grey, which
Mm-hmm.
is the same high school that Seth Rogen went to.
Uh-huh, yeah.
Um, and high school's all right. I got okay grades, enough to get to UBC Science, and I'm
Okay.
happy with that.
Mm-hmm.
And undergrad, my grades were like okay. I wasn't sure what to do. I did combined major in science. So I did essentially my degree was a combination of biology, physics, and earth and ocean science because I didn't want to do
Wow.
chemistry because I thought that would be too much.
Mm-hmm. Yeah, that's a
Um,
lot of science.
Yes, and I
Mm-hmm.
took it because I had no idea what field I wanted to go in. It was only like in my third year I really got hooked on to neuroscience, and
Mm-hmm.
then from there I knew I wanted to do neuroscience. Didn't know exactly what I wanted to do in neuroscience, but I knew I wanted to go in that field. And around that time I realized I wanted to go to graduate school, and so to do that I had to increase my grades.
Mm-hmm.
Um, and so I managed to do that. Although I still got this is, I guess, just a tip for anyone in undergrad.
Yeah.
I didn't realize for like biology or neuroscience graduate school, I should have, you read all the requirements, but when they look at your GPA, it's 300 level credits typically is most important, but
Mm-hmm.
they include credits in totally unrelated fields.
Oh,
So
wow.
at that time, I'd known I didn't want to do physics. And so my last physics course, I was kind of taking as elective. I was kind of interested, but then halfway through, I was like, it wasn't what I hoped it would be, the course, and I didn't need to take the course, and so I just barely passed. And I
Oh,
didn't
mm-hmm.
realize the physics course would affect my overall GPA for, applying to neuroscience. But I still managed to get in.
Mm-hmm.
But you don't have to have straight A's to get into graduate school,
Oh,
luckily.
okay. That's good to
Uh,
hear.
yeah. But that was stressful.
Yeah, that's for
Um, so,
sure.
so then I went to SFU for my master's. I got accepted at UBC, but the professors I had been talking to, I was talking to two of them. I chose one of them, and another lesson. Turns out he didn't have enough funding for another graduate student. He thought he did, but
Mm.
he didn't. And
Mm-hmm.
so then I went to my number two choice. He'd already accepted another student, and so I ended up going to SFU because of that.
Okay.
Um, yeah, so always make sure funding is nailed down.
For sure. Yeah.
Um,
Yeah.
but yeah, I went to SFU, ended up in a worm lab studying C. elegans, which is a small 1 millimeter long nematode. And I was studying actually in neurodevelopment. So
Oh, okay.
this lab studies axon guidance. So neurons send out long process called an axon to talk with other neurons. And that axon often has to go, you know, a long distance on like a cellular level, like even up to
Mm-hmm.
a meter. Um, and so it's got to navigate to the correct region of the brain. And so we looked at molecules that were involved in that navigation and we would knock them out. And then we would look at these axons and C. elegans under the microscope and we could see if they were going where they were supposed to or not. And if they weren't,
Mm.
then that molecule might be involved in axon guidance.
Wow, that's a
Um,
fascinating work.
yeah, not really related to learning.
Mm-hmm. Yeah.
And
But still,
so,
it's a connection between the neurons, and then the learning
oh
is
yeah,
involves the neurons. So I think
oh
in a way,
absolutely, and
yeah.
stuff like this could be involved in basic neurodevelopmental problems, um, and
Mm-hmm.
yeah, this work is often done in some of it's done in C. elegans, like the first gene that was found to guide axons, which is netrin, was discovered actually in C. elegans.
Oh wow. Uh-huh.
Yeah. So C. elegans is very useful for a lot of fields at the basic molecular level. Um, so from there, I actually wasn't particularly planning on going into another C. elegans lab. I
Mm-hmm.
looked at a bunch of labs at UBC. I knew I wanted to go back to UBC. My house is really close. My family home is really close to UBC.
Mm-hmm.
So and it's a great university. Like so, it just makes
For
so much
sure.
sense to
Yeah.
go back there. And so I looked at all the neuroscience professors' backgrounds, and I chose three labs I was most interested in. One of them happened to be Dr. Rankin, who
Mm-hmm.
is also a C. elegans researcher, and she does learning, which is why you're talking to me now.
Yeah.
And yeah, that happened to be the one that I got into. I knew her in the past. I volunteered in her lab. That helps. And so yeah, that's how I ended up to where I am, where we study simple learning, like mainly habituation and sensitization. Um, yeah.
Awesome. Awesome. Thank you, Nick. That's a very long and the great journey, let's say. So I think I have a few questions. For example, you worked in different labs, right? For example, back in Simon Fraser, now in UBC. What's the biggest difference you noticed between working in Simon Fraser and UBC?
So the universities are very different. UBC is sprawling, it's huge, and they can just keep cutting into the forest, which
Mm-hmm.
I don't like when they do it, but they can, and they
Mm.
do. So it's sprawling. Whereas SFU is on top of a mountain, and there's only so much you can expand. It's a very small mountain, also,
Mm-hmm.
and so they it's all jammed together. So the building is a maze, and also it's an old building.
Mm.
So like our lab, there's like asbestos in the walls. You don't want to go like digging in the walls, so that would be bad.
Mm.
Um, Although UBC does still have asbestos as well.
Yeah, yeah.
Like yeah, my lab here actually has some asbestos in the walls. So yeah, I guess that's kind of similar, but definitely worse at SFU. Um, Also interesting, the SFU lab doesn't have locks on all the doors.
Oh,
So like
okay.
UBC lab areas. So UBC will have like lab areas. So they have a building, and part of the building will be like a level or two will be labs, for example, and those require key cards usually to get in. But
Mm-hmm.
SMU does not have that, which is interesting. I guess they
Mm-hmm.
rely on the fact that it's just such a maze. Uh,
Yeah, for sure.
but the quality for neuroscience, UBC is definitely way better. They
Mm-hmm.
have way more professors. SFU is trying to advance in that direction. While I was there, some professors had just finally managed to get pushed through the bureaucracy to get a neuroscience specialization. So not even a degree
Mm.
program, but a specialization that like if you're taking a biology master's, you can get this specialization and say that have something on your transcript saying neuroscience, blah blah blah. And they're trying to work in that direction, and they've got a number of professors who work in neuroscience, but it's still something that the university is trying to strengthen.
Okay, yeah, awesome. So that's a very interesting difference that when it comes to the lock, right? That's something I wouldn't expect. Yeah.
Yeah.
Okay, awesome. So let's get into actually now dive into today's topic following onto your the current lab work. So we're talking about this basic, the very first form of learning, and which is happen to be habituation sensitization. Could you please more explain about what is habituation, what is sensitization?
Yeah, so these are very basic fundamental forms of learning that most
Mm-hmm.
people don't even wouldn't even think of or as occurring. Like it's not something like if you were trying to like back a couple hundred years ago trying to think of different types of learning, you probably would just ignore this. It wouldn't even come onto your radar. So habituation is the decrease in a behavioral response to a repeated stimulus. So if the feeling of your clothes on your skin is repeated over hours in the day, after a few minutes, you're going to, or even a few seconds, you're going to totally ignore it and you're not going to respond to it at all. Same if there's like a ringing siren, it's going to be really annoying, but you're going to stop responding to it pretty quickly. And this allows your brain to focus on stimuli that are actually important. Maybe you're trying to focus on studying for a test or paying attention to danger. Evolutionarily, this is really important, especially for humans where you've got all this concept for abstract thought,
Mm-hmm.
which can't be utilized properly if you can't actually focus. Um, and so habituation helps animals to do that. Sensitization is kind of like is the counterpoint to habituation, where a stimulus causes you to increase your response to future stimulus. And sensitization is a little different than habituation, where if one stimulus sensitizes you, you become sensitized to a broad range of stimuli.
Mm-hmm.
In the course, we taught you as your whole body is sensitized, but that's not quite true.
Yeah.
There's a lot of subtleties that they're still trying to understand, but certainly, if you get startled while you're driving a car, you're going to be more likely to overreact to stimuli that happen through the rest of the drive, for example. Yeah.
Mm-hmm. Yeah, I think I remember one the example on the sensitization that, for example, there is some kind of thing that touched their skin, and then you feel like it felt like a bug, and then for the next probably like 2 or 5 minutes, if you were very afraid of the bugs, you feel like oh every time something kind of like the weird touches you, you feel like bug is running around you. So that's kind of like
Yeah.
the example of sensitization.
Yeah, exactly. So if you've been stung by a bee or you just like had like a scary bug on your hand, you flicked it off, just like the
Mm-hmm.
feeling of the wind on like your skin is gonna like make you look down and like, is there another weird bug on me? Yeah, that'd be an
Mm-hmm.
example of sensitization.
Okay, awesome. So Nick, we're talking about a lot of these C elegans, right?
Hmm.
So why we choosing the C elegans and then why we start this learning from this one nematode? Why is it important, this nematode, C elegans?
So my professor, my supervisor, Dr. Rankin, started the field of learning in C. elegans. And it was by happenstance. She was actually planning on studying a sea slug aplysia,
Mm-hmm.
but the you have to aplysia is you can't grow them in a lab, so you've got to fish them out of the ocean. And her shipment of aplysia did not come in, and so she
Mm-hmm.
chose C. elegans. Because there was other labs at UBC working on C. elegans, and she'd heard a lot of great things about what you could do with them, and they're small, so they fit with the equipment she had in the lab. Um, and so with C. elegans, what you can do with learning, you can study a lot of simple learning paradigms like habituation, sensitization, even classical conditioning. There's a lot of studies in C. elegans where you train animals on food in the presence of a cue, and that gives a positive association with a cue, or you train them off food with a cue, and then they associate that cue with starvation, and they'll avoid it.
Mm-hmm.
Um, and of course, you can do that in other animals, but what you can't easily do in other animals is interrogate the genes involved in learning. It's getting
Mm-hmm.
better in mice. Like the genetics in mice that they can do nowadays is incredible.
Mm-hmm.
But for decades, if you want to do quick genetic research to actually find out what gene was causing your behavior or the genes
Mm-hmm.
were affecting your behavior, C. elegans was the best way to go, and it still is. But mice probably catching up, I would
Mm-hmm.
say. But it's still, for example, in my lab, we often, like a graduate student, will investigate dozens of genes
Mm-hmm.
for whatever behavior they're looking at. And so you can discover a large number of genes affecting your type of learning. And then you can also get, because you're using silicons are so small and you can test so many animals um, quickly, you
Mm-hmm.
can this lo- this large N is like the shorthand for large number of experimental animals. This large N allows you to increase the power of your observations. So whereas a
Mm-hmm.
mouse study might have like 8 animals, and you'll have, maybe initial response and then final response, and there's like a big error bars for each of these bar graphs. With C. elegans, the error bars are much smaller because you have much more animals to choose from, and
Mm-hmm.
then you can find more subtle variations in behavior that you'll miss in most other animals. For example, our labs discovered that habituation, so I mentioned habituation before, the simple form of learning. When
Mm-hmm.
you, in the past, habituation was treated as your response habituates and it's all one process. So habituation is a single process that's decreasing this response. But it turns out that's not true. Different components of a response can decrease at different rates, or some might not even decrease at all in some situations and will decrease in others. And we found genetically that some genes are involved in habituating particular response components. And so by studying this in C. elegans, we identified different genetic pathways that are controlling different aspects of habituation. And this shows even the simple form of learning is not a single process that's
Mm-hmm.
on or off. It's a much more modular process, which allows behavior to be much more dynamic and adaptable. So you're not just turning a response up and down, but you're actually molding the response to what this new environment you're in requires. Yeah, so those are some of
Okay.
the benefits for studying learning in C. elegans.
Mm-hmm. Yeah, that's a great point. And I think one thing I really remember was that because elegance it's very simple, right? It's very small, very simple. It's
Yes.
the the neurons, they are a very small amount, and also the genes, very small amount. So it's very easy to make a research in it.
Well, so yeah, the neurons are very small. There's 300 neurons. Genes has actually got, I think, like over 20,000 genes. So it's
Oh,
got a
okay.
similar number of genes, I think, to humans. And you'll find that in a lot of like it's hard to guess the number of genes based on how
Mm-hmm.
simple the animal is,
Mm.
interestingly. So it's got a lot of genes, but the whole genome has been sequenced for decades. And so that also is a big boon.
Mm-hmm. Yeah, it's apparently the first ever the living species that the genome was fully sequenced, right?
Yes, it was, and a lot of the people who worked on sequencing the C. elegans genome were spearheaded the sequencing of the human genome. So they
Mm-hmm.
brought their
Yeah,
expertise to humans.
that's awesome. That's awesome. Okay, so now the first form is the we talked about this habituation sensitization. So let's get into now the conditioned behavior. So the way we behave in many different environments, we usually just in conditioned responses to different the stimuli, right? So let's start with the classical conditioning. So what is classical conditioning and how it works for us, or what are the studies that started this?
So classical conditioning is when an animal, it's essentially when an animal's pairing two stimuli. It's
Mm-hmm.
animals notice a relationship between two stimuli. For example, the classic experiment is Pavlov with his dogs, where you paired a conditioned stimulus, a bell, with an unconditioned stimulus, food, and the animals learned that the bell predicted food, and so they would start salivating not just to the food but to the sound of the bell. Um, and so that in basic sense is what classical conditioning is.
Mm-hmm, mm-hmm. So yeah, we talked a lot about the classical conditioning. So why is it important for the species to have this classical conditioning? Why is it very important?
So classical conditioning is another quite simple form of learning that you're doing
Mm-hmm,
all the time, and it happens mostly unconsciously. At least that's my understanding. And this is going a little further from my expertise, but you're doing it constantly. And for example,
mm-hmm,
maybe something's your favorite color because something in your past when you were a kid, something really good happened and there was a lot of blue around. Like that could
mm-hmm.
mean that becomes your favorite color. Similar for why you might hate a color. So all
Yeah.
these associations are you forming, and you might not even remember how you formed this association, but your brain is constantly forming like this particular stimuli, when this is in the environment, something bad always happens. And
Mm-hmm,
so you'll get like really anxious in this particular circumstance, for example. Um, so these stimuli let you, these associations let you predict what's going to happen in your environment and act accordingly. And it can be, again, it can be an unconscious prediction. For example, drug users, they become their body becomes gets ready for the drug when they're in a specific when they're in the area that they usually take the drug,
Mm-hmm.
and the body will realize that this area predicts drug use, and your body will compensate for the effects of the drug in the opposite direction so that the drug won't have quite as bad as effect on your body. And so your body is predicting what's going to happen for you. Again, no conscious thought is required to help
Mm-hmm.
improve your survival. And so if you do take this same amount of drug in an area that your body isn't ready for, you might have an overdose because your body hasn't predicted that you're going to take the drug and is not ready for the volume of substance that you usually consume.
Mm-hmm.
Yeah, so that's an obvious way that it helps you survive.
Yeah, yeah, that's for sure, right? For example, like the classic conditioning is, I think one of the ways habitual desensitization, all these unconscious, like these experiences helps us to focus more on important stuff and free us from this unnecessary. For example, once you learn these things, predicts that thing, you don't need to focus on and figure out what's coming next, right? Because you just know it, classically conditioned in you, so you don't have to focus on it. You can focus on different, more important stuff, right?
Yes. And
Mm-hmm.
it's important to note that these, for example, especially sensitization and classical conditioning can also be maladaptive in certain situations if they get too strong, if the association gets too strong,
Mm-hmm.
or, for example, PTSD often involves hypersensitization. So, yeah, as with most things, they can go astray in disease.
Mm-hmm. Okay, awesome. So, we talked a lot about also like therapeutic stuffs that involves classical conditioning. As we know, one of them very famous thing we learned in the class was the systematic desensitization, right? Could you please
Mhm.
talk more on this one?
Yeah, so this is really interesting, and they still use it now. It's a very effective therapy. So, say you're afraid, you've got some sort of phobia of, let's say, spiders.
Mm-hmm.
Um, so, systematic desensitization allows you to relax they teach train you to relax in the presence of stimuli that cause you this anxiety or fear. And this pairing of this relaxation with this previously feared stimulus counter conditions and alters your association, your classical condition association with this stimulus so that you now no longer have this heightened fear and anxiety response to it and have a more natural response to spiders. And they do this incrementally. So at first you might just be told to like think of a spider and try to feel relaxed. And then they'll like a drawing of a spider and eventually they'll bring a spider into the same room as you or even have it like crawl all over you, I think.
Mm-hmm.
That's like the final stage. Um,
Mm-hmm.
and so that can desensitize you to these stimuli that you are afraid of to unhelpful degree, let's say.
Mm-hmm.
um, and you can do this in, I think they do this in PTSD as well, where someone will have a particularly traumatic event, for example, and they will try to desensitize you to stimuli associated with that event.
Mm-hmm.
So maybe the event was associated with a splash of water on your back. Like I read a story about that. And so they would try to desensitize someone so that whenever they felt water on the back, they didn't freak out. Then it's fine. It's going to be okay. And so they would try to slowly desensitize people to that particular stimulus.
Mm-hmm.
Um, and actually, it goes a little further. So when you use this treatment in PTSD, this systematic desensitization,
Mm-hmm.
you it's effective at um, getting people to no longer feel the same stress or anxiety about the particular stimulus associated with that like episodic memory of the
Mm-hmm.
trauma. However, it often doesn't just cure the PTSD. This person might still have generalized anxiety or other kind of stress problems, and they found in mice that when they stress these mice out, they often have non-associative problems. So this would be sensitization.
Mm-hmm.
So it's not just a classical conditioning association between the trauma and stimuli related to the trauma and fear. it's also, it might have actually altered your sensitization response. So you might
Mm-hmm.
be generally more sensitized. So this would be like generalized anxiety. Or you might be more susceptible to sensitization. So the rate of sensitization that you have is actually might be different. And these things, generalized sensitization and rate of sensitization, they might sound very similar, but they've
Mm-hmm.
actually found in rodents that they're actually controlled by different parts of the brain and that they don't actually correlate with each other. So
Oh
someone with
wow.
PTSD might have a more generalized increase in sensitization problem, or they might have a more heightened sensitivity where something quickly becomes sensitized for them. And those are
Mm-hmm.
different problems. And so that's something that I went to a conference last year and they were talking about that. It's really fascinating.
Mm-hmm.
That's another way that, I guess that's an even more modern approach that they're taking and using classical conditioning to treat PTSD.
Mm-hmm. Yeah, that's very interesting because like when the thing we were talking about was like the desensitization, it helps with the PTSD and a lot of the exposure therapy and all this stuff. But there's a two types. So one is generalized into a different, as you said, the one is more focuses on the stimuli, the response, how you respond to the stimuli. Another one is more to sensitization, right? So
Yeah,
it's a very interesting for
yeah,
sure.
exactly.
Okay, so I think on this one, there's also another one we talked about in the class that we talked about this aversive counter conditioning. So could you please get into more in aversive counter conditioning? What is it and how it's related to classical conditioning?
Okay, so aversive counter conditioning is so in the previous example, you're trying to pair a positive um, response with a stimulus. And here you're trying to pair a negative outcome with the stimulus. So
Mm-hmm.
you're trying to make something aversive that wasn't aversive in the past. And so why would you want to do that? Well, they've tried this for people with drinking problems or smoking problems. Um, with, for example, they'll pair nausea with
Mm-hmm.
drinking. So people have people drink alcohol, which includes some sort of chemical that will cause you to throw up or feel very nauseous. And they'll force people, well, they're not forced, people will agree to do this, but they will, in one of these sessions, they'll require people to drink a large amount of alcohol flavored substance with this nausea to really promote this association between nausea and alcohol. And the idea is that so they're less likely to drink in the future because they've got this negative association with it now. Like for example, if you, I don't know, joined a book club in the first session in the book club, everyone made fun of how unread you were, and
Mm-hmm.
you had an awful experience, you've got this aversive association now with book clubs, you're a lot less likely to do that in the future. So that's the idea. You're now pairing drinking with this aversive stimulus.
Mm-hmm.
This doesn't seem to be as effective as systematic desensitization is for phobias.
Mm-hmm.
There is a strong recurrence rate, although this seems, I haven't read this studies, but it seems like maybe topping up these sessions every so often makes them more effective.
Mm-hmm.
Um, but yeah, that's definitely something that's been tried to modest success.
Mm-hmm. For sure. So, as you talked about, these are saying not making that it's not as effective as systematic desensitization. So, what's the underlying causes for these are not as effective as the treatment for the phobia?
Hmm, why is it not as effective? I don't quite remember. I'm not sure. Let me see if there's anything in the lecture notes. Hmm.
I
Why is
think
it
if
not?
I don't remember wrong, I think it was the one of the things that were making it was because of the context. For example, like you made this context learning. For example, when you are smoking, you meet with the same friends who are also used to smoke. So basically, like you make a
Right.
treatment, so get it back to your the context, and then it kind of like also creates the
Yeah.
cues.
Okay. I see. Yeah. You've got. Yeah. In the case of yeah, drinking or smoking, you've got all these other associations working against you. For example,
Mm-hmm.
yeah, you always drink with this group of friends, and there's also social pressure, of course, in that case as well. And so
Yeah.
yeah, stuff like that will make it much harder for it to be effective,
Mm-hmm.
and slowly eroded over time. Yep, yep.
Mm-hmm. Okay. Awesome. So I think now we can get into instrumental conditioning. So there's a two very big conditioning. One is, as we talked, classical conditioning, and then now could you please tell us more on instrumental conditioning and what is it about and what's the difference between the classical conditioning and instrumental conditioning, how it's different?
So classical conditioning, you're pairing conditioned stimulus with an unconditioned stimulus, and because of that pairing, you're reacting differently. In this case, you're pairing positive or aversive outcomes with a behavior, and this
Mm-hmm.
will increase or decrease the rate of this behavior. So if you pair um, application of food to when an animal jumps, it's going to jump more often if it's hungry to get the food. But if you pair jumping with a shock, the animal is going to stop jumping because they don't want to get shocked. Yep.
Mm-hmm. Mm-hmm. Awesome. So there's a very famous experience on instrumental conditioning by Dr. Skinner, right? And operant chamber. Could you please explain a little bit about like Dr. Skinner's experiment and then why it was so instrumental for the science?
Yeah, so he developed the operant chamber or the Skinner box, where you put a rodent, although it can be other animals as well, in a box with a lever, sometimes a light above the lever, and some sort of delivery mechanism for food pellets. And so the animal will learn either on its own, but more likely you try to help train it that when it presses the lever, it will receive a food pellet. And so once it makes this association, it will press this lever whenever it's hungry to get the food pellets.
Mm-hmm. Yeah, so basically, like he made the class and the instrumental conditioning very easy to make the research and experiment in very short time. For example, we talked
Yes.
about Maze Runner and then a lot of different kinds of stuff, but it takes a long time. Also, recording is hard, and there is many other problems. But as he introduced the operant chamber Skinner box, and it made it much more easier and faster, right?
Yes, and it was much easier to another big advance is the food delivery mechanism. They linked it to a cumulative recorder, so it'll just a stylus on a piece of paper that
Mm-hmm.
will and a piece of paper that's constantly running through over the stylus, and the stylus moves whenever the animal presses the lever and gets food,
Mm-hmm.
and so you can automatically keep track of the animal's learning as it presses the lever more and more often. That's an increase of learning in this case, and so you can actually automate. Well, one, you can automate recording of the learning, but also you have now a really easy measure of learning throughout the a continuous process,
Mm-hmm.
instead of the trial-based processes that were often manual, like the maze running before.
Mm-hmm. Okay, awesome. So now let's get into the reinforcement learning. So why reinforcement is very important for learning and it helps us to learn even more. And what's important when it comes to reinforcement learning? Is there any important things we need to look out?
So reinforcement is key to a lot of forms of instrumental conditioning. Um, and you can see this in your own behavior. Like if well, maybe not your own behavior. A lot of people don't do this to themselves, but like teachers will try to reinforce good behaviors that they want you to keep doing. And reinforcement is can be really good to provide external reasons for someone to do a behavior that you want them to do. But you can also do it on your own. Like in my case, I needed during COVID to go running because there
Mm-hmm.
wasn't any. I usually play soccer for exercise, but all that was canceled during the height of COVID. So I needed to
Mm-hmm.
get running on my own to keep fit, and I really hate running. Um, so I needed to reinforce this behavior, or else I was going to stop because there's been plenty of times where I tried to get running to get fit, and I just can't because I hate it so much.
Mm-hmm.
but I reinforced it with I got to have some cookie dough. Every time I ran, I would have some cookie dough afterwards. Now, I wouldn't recommend this if you're trying to run to lose weight. This would be counterproductive, but that's not what I was doing. And so
Yeah.
this, I did this, and now I don't have as, just even when I think about running, I don't have as much of a negative association. And I kept through it through COVID, no problem really. So that's a way that reinforcement can really affect your behavior, and I was surprised how effective it was actually.
Mm-hmm.
Yeah.
That's awesome. So when you're saying that you're reinforcing right after you're running, so when you are reinforcing yourself and then try to like create this habit, right? What's important things to look out?
Yeah, so you want to make sure the reinforcement is applied right after the behavior you're looking for. So even though I knew what I was reinforcing myself for, this kind of learning is again, it's very deep in your brain, like in a lot of these unconscious parts that you don't even realize how much it influences your behavior. And so these parts of your brain are making the association. And so I wouldn't make such a strong association between this beneficial thing and running if I, for example, got the beneficial cookie dough the next day. My brain would not be able to make that connection, even though logically I can. That
Mm-hmm.
kind of emotional feeling you feel towards running and how that changes, that wouldn't happen, or at least not as much. And for example, if you're trying to train a pet, the same thing. It's really important to link in time. So time is very important. All these learning assays, but it's really important to link in time the training and the reinforcement.
Mm-hmm.
The animal is not going to figure out what you're punishing it or reinforcing it for if it's not right after. And even then,
Mm-hmm.
it's going to take a while.
Okay. So yeah, exactly. So for example, like the when the teacher, I remember one case that Dr. Rankin was talking about. He's when she was a teacher, she was talking about this one kid who was very kind of like the messing around and then like the not doing the work he's supposed to do. And then Dr. Rankin was talking about like, okay, you can go out and whenever you want to study, you can come in. And then Dr. Rankin, what she was focusing on was whenever he does study, right? He actually does study. Whenever he does something good related to studies, he actually keep saying, reinforcing things, right? Oh, you're doing great. You're awesome. And basically, like as she was saying, was catch him when he was good, right? And then immediate reinforcement, and which is helps him to be more the increase that
Yes,
behavior, right?
and this is often a problem with parenting and teaching as well.
Mm-hmm.
You often forget to reinforce people when they're doing something good.
Mm-hmm.
You might think, oh, that's great, they're doing that, but you don't actually reinforce them. It's only when they do something bad that you'll punish them. But that's
Mm-hmm.
actually less effective at changing the behavior than reinforcing them when they're doing something good. It's also important to realize what's reinforcing and what's punishing. That will be determined by the individual often. Some reinforcements and punishments are not universal. In the case of the kid you were talking about, attention was reinforcing. And so when you get angry and punish him when he's doing something you don't like, it's not always interpreted as a punishment because he's getting attention. And so you actually end up reinforcing the behavior you're trying to end.
Mm-hmm.
So realizing what's actually reinforcing or punishing for the individual you're trying to change their behavior is really key.
That's a great point. So I think now we talked a little bit on this one, the punishment, right? So we talked about the reinforcement, which is increasing the desired behavior, and we used it in a lot of animals, in a lot of in cases. And now let's get into the punishment. Punishment is also widely used in the many cases. So what's very important in punishment is important for the learning or the decrease in the behavior.
Yeah, so if you when someone or an animal makes a behavior and then you do something that punishes them afterwards, whatever that might be, they will decrease that behavior. However, it tends to be less effective at decreasing behavior than reinforcement, positive reinforcement. Um, and for example, they might just not do the behavior when you're around, and as soon as you leave, they will do it again because they still want to do the behavior. It's just they realize if they do it when you're looking at or when you notice, they'll get punished. So it often doesn't cause a change in their internal motivation.
Mm-hmm.
It'll change their actual behavior they represent, but the depth of that behavioral change is often quite shallow.
So it's basically like it's not really meaningful change. It's just that because you are looking at them, the person is doing that, and then if you are going out, and then the kid is not doing that anymore, doing the behavior, right? So it's very important to intrinsic motivation, and then
Yeah, I wouldn't say it never changes
mm-hmm,
intrinsic motivation, especially if you pair the punishment with an explanation for why they shouldn't be doing that. Or obviously, you can't do it
mm-hmm,
with animals, but with children, for example, you can pair that with an explanation.
mm-hmm,
Then sometimes that can actually, the child will be like, "Oh, this sucked," and there's actually a reason that I shouldn't be doing that, and so it can
yeah,
change their behavior.
mm-hmm,
But yeah, positive reinforcement when they're doing other things that aren't that is more effective at getting them not to do that
mm-hmm,
often.
mm-hmm,
Um,
for sure. Okay, so when it comes to punishment, there is a very important thing to look at is how strong the punishment is, how the is strength of the punishment is also very important for it to be more effective, right? Can you please talk about how we can choose the right amount of the punishment?
yeah, so this is got some interesting animal studies on this topic. I think
Mm-hmm.
it was in birds. They would shock the animal when they press a lever, and if the shock was high enough that it would stop their lever pressing behavior, then they would behavior would remain suppressed even if you decrease the level of the
Mm-hmm.
shock. But if you shock them, but not at a high enough voltage to stop the lever pressing behavior, as you can increase the shock quite a bit before the animal actually stops this behavior. Like many times the initial shock value that would have stopped the behavior. And so this mild punishments can actually make a behavior resistant to punishment.
Mm-hmm.
Um, and so if you are going to be doing a punishment, starting at a mild punishment that doesn't have much effect is actually counterproductive.
Mm-hmm. So that's why it's very important to like choose the right amount of the punishment. Otherwise, it's make it more resistant to the punishment, and then further punishments are not as effective, and then it might even resistant to it, and then punishment can never work again for the person, right? So
Yep.
also, if you choose very high amount of punishment, it can be damaging to the person, right? It can be traumatized, or it can be
Yes.
even on the animal. It can damage your animal, right?
Yeah, it can have a lot of adverse side effects that extend well beyond the particular situation that you're punishing. And again, it can be its own traumatic experience that they get PTSD from if it gets really bad. So, yeah,
Exactly.
yeah.
So it's very important. So I think you just briefly mentioned about this study on like explaining is very important in terms of the punishment. So as a humans, the language is a very important tool as we communicate with each other and then as a social species. So why can you explain more on this one and why the language and explanation is important? And then can you get into more depth into the studies related to this?
Okay, so in those studies, I think they had kids and they had different toys laid out before them that they could play with, and they would punish some of the kids if they played with the more shiny, newer toy, more complicated toy often, and they would punish them and they say don't play with that toy, and then they would walk away and see how long or how it took for the kid to play with the toy again.
Hmm.
And so if you just punish them, it was effective, but only to a certain degree. They would stop playing with the toy again at a certain point. But if you punish them and say, hey, this toy is too complicated for a child of your age, or something, some explanation like that, the change in the behavior was much more dramatic, and many of them didn't play with that toy even over a long time period. Um,
Hmm.
yeah, so the explanation can have a large effect on behavior, even if the explanation isn't that great. Like in that study, that doesn't sound like a great explanation that
Yeah.
as a kid might have been like, oh, it's too complicated for me. Sure. Like it's not a great explanation, but any explanation is better than no explanation.
Yeah, for sure, right? So I think they're not just saying no. It's instead of just saying no, you gotta explain
Exactly.
why you shouldn't do this, and then it helps the kid to understand the reason behind it, right?
Exactly.
Okay, awesome. Also, you just briefly mentioned about the side effects of the punishment. So we talked a lot of about lot of experience on the what could be. We kind of encouraging this positive reinforcement in the replacement of the punishment to change the modify the behavior. So part of the reason is because this punishment has many side effects onto it. So could you please explain what are the possible side effects on the punishment?
So one possibility is the punishment can you're trying to like punish a particular behavior and stop a particular behavior. For example, someone in school is talking with their friend while they should be working, and so they get punished. But this often can lead to generalized effects with you're not just reducing the behavior that you want, but you're causing a more generalized negative change in the person, where they generalize this punishment to school more specifically, and they start having a negative experience with school and don't want to go to school as much. Instead of what you wanted was just don't talk while you should be doing your homework. So these kind of generalized effects can be quite pernicious and can be totally counterproductive to what you were trying to do. It can also, there's animal studies with rats where when you punish them a lot, they're more likely to be aggressive with other rats that they're housed with, um, to the point where they'll eventually kill each other if you really want to get barbaric about the experiment. And so punishment can increase aggression. It can increase a lot of negative emotions like anger. They can be angry at you. Um, you would want them to be, self-critical and, oh, I don't want to do that, but that's not often how it is. So yeah, there's all these punishment can have all these wide range of effects that you cannot control. Only the person being punished actually can control.
Yeah, for sure. There's a one example that Dr. Rankin and you just mentioned was that if you get punished at the school and many times, eventually you don't want to go to school, and
Exactly.
even it can generalize into the learning. You don't want to learn anymore, or
Yep.
you hate books anymore,
Yeah,
right?
you hate
And
math or whatever course that it was. Yep. Yep.
exactly right. So it has very diverse the effects related to the punishment. So I think the next one I want to mention was that we talked about the punishment. So there is also punishment when there is not actually punishment. So when we're talking about reinforcement a lot, and sometimes it can become a punishment. Could you please talk about extinction as a punishment?
Yes. So if you're reinforcing a behavior and then you suddenly stop reinforcing it, and the animal has learned to expect a particular reinforcement, then that is behaviorally doesn't, and possibly in the brain, there's not much distinction between that and punishment. The animal will act more aggressive, angry, frustrated, and will look as if it's been punished. And in reality, that makes sense. Like, you can, for example, with a child, you can punish them by not doing something or taking something away that they would expect to be there. Um, and so this all of these types of learning that we've talked about are involved in animals' expectation of an outcome. And when you're removing a positive outcome, it's much like adding a negative outcome. The change in outcome is quite similar, actually. Um,
Mm-hmm.
so yeah, often treated like a punishment.
Okay. Yeah, it's very important to also like the reinforcement and then how much you're reinforcing because like the once you start very high reinforcement and if you decrease the reinforcement it actually no longer reinforcement it actually like decreases the value of the reinforcement and it becomes a punishment for the person, right?
Yeah, exactly. So if you, it's a drawback for reinforcing too much and without changing like the animal's or person's intrinsic motivation. If you take away the reinforcement now, then they'll stop doing the behavior sometimes.
Mm-hmm.
So in that case, you want to measure out the reduction in reduction in reinforcement, so that it's not just all or nothing. It takes a series of step decreases in reinforcement. Yeah.
Mm-hmm. For sure. Also, I remember one of our last classes we were talking about this token economy, how we make the enforcement as effective as long as we want. So how could you please explain more and how we can make this so reinforcement can get out of like can become ineffective very quick. So how we can continue the lengthen the reinforcement.
Yeah, so you can use tokens, points, these are secondary reinforcers, money.
Mm-hmm.
So
Mm-hmm.
the typically reinforcement you're taught it as like some primary reinforcer like food, how like shelter, something that immediately benefits you.
Mm-hmm.
However, humans are easily capable of abstract thought and it comes naturally to us. So you can easily train people or reinforce behaviors with secondary reinforcers. So this happens all the time to you with like points on specific rewards cards company use, and they'll reward you for the behavior of shopping with them by giving you points, which are secondary reinforcers, which you can then use in the future to get primary reinforcers.
Mm-hmm.
Um, and so this is a way to quickly and effectively pair a behavior with positive reinforcement.
Mm-hmm. Okay, that's a great explanation for sure. And Nick, so on the next one, I think I want to talk. This is a very much the talked with in the class, very serious topic, and then there might be right or wrong. It can be very different for everybody depending on their opinion. So, what do you think about the punishment in humans? There is a the way that the punishment is the only way to get out of the situation to change the behavior. What are those situations could be, and then what are the studies and the previous experiences show?
Yeah, so we've talked about all these principles of reinforcement, punishment, when is it effective, when is it not effective. And if you apply this to, for example, our criminal justice system, it doesn't line up very well. So
Mm-hmm.
the punishments are delivered much later than the actual behavior. They often start very mild and then creep up, which of course we've just talked about that makes it resistant to punishment. But at the same time, you don't want to. It's a society we've decided we don't want to slap large penalties to minor offenses. So we've moved away from that, and we didn't do that like we weren't like draconian or anything, but we've moved even further away from that in the past decades.
Mm-hmm.
And is that a good thing or not? I'd have to think about it more. It's really these are kind of decisions that you don't make lightly,
Mm-hmm.
and there's a lot of negative consequences to um, doing a really harsh punishment. So, for example, prison time for an offense that normally wouldn't get prison time. If you add prison time, you're going to have all these other negative effects. You're going to probably decrease the behavior, sure, but you're going to also cause all these effects to this person. For example, taking them out of their community for a period of time. They might lose skills, ability to actually get a job, etc., etc. Um, and so definitely programs to reinforce working, like ways to get people with felonies and other bad marks on their record to be able to get reintegrated into the society and work is probably the most effective way. Um,
Mm-hmm.
because right now, again, I'm not an expert on all the laws we have, but it's definitely harder for people who have had these black marks on their resume to then do the things we want them to do. It's actually harder for them to do that, and they already know how to do the bad thing. So
Yeah.
it's definitely can cause more recidivism. So somehow reinforcing the things we want them to do is going to probably be the way to go because we're never going to decrease the um, the temporal contiguity between behavior and punishment in our criminal system because you have to have trial and it takes time.
Mm-hmm. Yeah, for sure. Exactly. And so there's a one flaws of the laws and then how the punishment is not working as it's intended, right?
Yeah.
Okay. I think one of the things I also wanted to dive into was that if there is a positive reinforcement is not be able to use because the behavior itself is not showing up, because if the desired behavior is not showing up, it's very hard to use positive reinforcement, right? Because when the desired behavior shows up, you use positive reinforcement to reinforce that behavior. But the person is not be able to do this positive, that desired behaviors, and then keep doing the self-destructive behaviors. So what I'm gonna talk about is the self-destructive behaviors in the children with autism, and then these kids are on autism spectrum disorder, and then on the very high end, they're very much the very heavy autism, the people with the
Yeah.
very heavy autism. And what do you think about this? And they used to be back in a and this one study and the one case that was they used electrical shock to decrease the behavior of the self-destructive behavior in children with autism. And could you please talk more on this one? Is it?
Yeah.
Mm-hmm.
So as I'll explain, and I haven't looked into this beyond what we discussed in the course, so I haven't like
Mm-hmm.
read these studies myself and looked up what the laws are currently. But essentially, in the 90s and before, there was these therapies where you had these particular type of, well, autism is a spectrum, and a few people on the spectrum have this unfortunate condition where they will just bludgeon themselves to death eventually, or
Mm-hmm.
severe injury. So this kind of extreme self-harm behavior, and it turns out you can control this behavior with really mild shocks when they self-harm. Um, and the this was effective, and then the government of some places, some provinces banned it because obviously people had a negative reaction to hearing about children being shocked. In psychology, shocking people has a very, shock therapy comes to mind. It's a very negative connotation. They use it all the time on people with schizophrenia, and it was not effective at all. However, in this
Mm-hmm.
case, mild shocks were very effective, apparently, at treating this condition. And so in this case, I would be very much for it. Again, I haven't read the data, but from what we learned in course, I very much support it because it is a mild shock. Like, for example, the parents felt the shock themselves. They knew how
Mm-hmm.
powerful it was. It's quite mild and it could totally stop the behavior, and the child could live their best life, go swimming, enjoy themselves without, bludgeoning themselves so they have no ears anymore, for example. Like, that's how bad it is. Um,
Mm-hmm.
so this is a case of, I would look into this before I would have a strong opinion either way, but that
Mm-hmm.
includes a strong opinion of saying, oh yeah, I want this banned. So you should actually look deeper, and if there's this therapy that doctors are for, not just like quack doctors, but actually like societies are for, it's probably for a reason, and you should probably educate yourself more before you take a strong stand one way or the other.
Mm-hmm. For sure, Dr. Rankin was mentioning on this one, she was saying the critical thinking is very important. If you make a decision, you need to educate yourself on on the matter that you are making decision on, and therefore your decision might have changed depending. For example, in this situation, right? Then many people say, of course, punishment is bad. Why would you punish the person and shocking the kid? What are you doing? But in this case, for example, positive reinforcement was very much impossible to use. So that's the only way to get out of the situation, and probably in this case, it worked. Luckily, right?
Yep, and yeah, and there's actually in I'm TAing right now Psychology 101, so
Oh,
introductory psychology, and there's a phenomenon called Mount Stupid. I don't know if you've heard of it, but essentially the what people who know the least about a topic are most confident on their beliefs.
mm-hmm.
However, most things are more complicated than they initially seem, especially if different groups of people are arguing about it. Typically, one side is not going to be like 100% right and the other side is 100% wrong. It happens, but it's typically more complicated. And
Mm-hmm.
so, people are most confident when they know very little about a topic. That's Mount Stupid. And then as you learn more about the topic, you get less and less confident, and then until you become an expert where your confidence starts going up again, but it never reaches the heights of Mount Stupid. So
Mm-hmm.
the people who are most confident are not experts about a topic, but it's the people who don't know anything at all.
Mm-hmm.
Um, and yeah.
Yeah, that's for sure. That's a very true. Before I enter UBC, I thought I was smart, and then after entering the UBC, I know very much that I'm stupid.
No, no, no.
So
Don't worry.
yeah,
You'll get out. You're in the valley of despair now, but you'll as you learn more and get closer to the expert level, you'll get more confident again.
yeah, that's for sure. So yeah, awesome, Nick. So I think those are the topics that I wanted to cover in today's episode. So now let's get into what's your future plans and projects you're looking forward, and how do you see what's your like the last the very much your goal in the future and the projects you want to do in the future.
Like as far as my research or like career
Yeah,
goals.
yeah. For example, let's start with the research. What do you want to do in the future, and are you working on some specific project, and what's your next project might be?
Yeah, so my work very much looks at the well, it's on the basic learning, so habituation and C. elegans. But my particular project is involved with the timing of stimuli. I mentioned a little bit
Okay.
earlier, but timing is incredibly important to all these forms of learning. Your brain is incredibly sensitive to the timing of stimuli. And it turns out for habituation, if you space stimuli further apart, you habituate slower. However, you remember that habituation longer. So if you habituate to stimuli that are 10 seconds apart, you'll habituate quickly, but after 5 minutes, your response will have recovered to naive levels, essentially.
Mm-hmm.
However, if the stimuli were 60 seconds apart, so a minute apart, and then you habituate, you don't habituate as much, but 5 minutes later, your response will still be fully habituated. Um, and so this timing of the stimuli, subtle difference in timing, is really important to the memory and learning process of habituation. And so I'm studying right now genes that seem to be acting in habituation at particular time intervals between stimuli. So we've got some genes that seem to only act in habituation when the interval is 60 seconds or longer. And
Mm-hmm.
at shorter intervals, they aren't very important in habituation. But at longer intervals, you hardly habituate at all if you lose these genes in the
Mm-hmm.
worms. Um, and so we think that again, this is another example of this simple learning process actually being a mosaic of different processes acting together to get the unitary behavior you see at the end and that you're measuring. But it's
Mm-hmm.
actually this behavior, this learning behavior you're measuring is actually the result of a large number of different learning processes happening in the inside these neurons and between these neurons
Mm-hmm.
that combine to this final behavioral output. And I'm trying to discover these processes that are associating these stimuli over these like longer intervals. And depending on where my project goes, this might relate to plasticity mechanisms that can um, bridge stimuli that are occurring minutes apart, for example,
Mm-hmm.
which is something I find interesting because most of the study on plasticity mechanisms is involved, for example, you've probably heard LTP and
Mm-hmm.
LTD.
Yes.
I think we talked about briefly in the course,
Yeah,
but essentially,
long-term potentiation, right?
long-term potentiation, it's been studied in for decades by
Mm-hmm.
hundreds and thousands of researchers, which is great. But long-term potentiation only involves association of stimuli that happen right after each other. So
Mm.
a neuron's receiving two inputs right after each other. It can associate these inputs with LTP. But as we mentioned, I'm not sure we mentioned actually in classical conditioning, though, in the course we talk about what's actually important for classical conditioning is whether something predicts the other, one stimuli predicts the other stimuli, not whether they occur at the same time.
Mm.
And
Mm-hmm.
often it can predict these stimuli. It's going to happen 5 seconds later, 10 seconds later, 20 seconds later. So they're not connected in time at all sometimes. And
Mm-hmm.
so I think a lot more needs to be known on mechanisms in the brain to associate stimuli that happen further apart in time.
Mm-hmm.
And right now, there's not a lot of mechanisms for that. So I think it's a
Mm-hmm.
really interesting field. And my stuff is loosely related to that in habituation.
Awesome, awesome. I think I remember the one of the things that I think might be similar to your work right now is distributed training. So, for example, through distributed trainings, it can help you to memorize it even more and then the learning process can they can stick with you longer.
Yes, so the distributed training is an effect that, again, we see it in habituation, but we see it in pretty much any kind of form of learning that's been measured in any model organism. If you train all at once, you're not going to remember it very well. If you space it out over time, you're going to remember it better. For my work, I've been theorizing why that happens because
Mm-hmm.
it's a very well-known effect, but why does it happen
Mm-hmm.
is still obviously it's more of a philosophical question, but why did we evolve that way? So for my work, I've been thinking if something's happening all at once, so you've never seen the stimulus before, and it happens to you all at once in this one big training session.
Mm-hmm.
It's only ever happened to you at one point in your life, and so what are the odds are that you're going to need to remember this for the future in your life? Well, not as high as if it's been distributed over different time periods, in your life. Then it's much more likely that it's relevant for you to remember it in the future. And so I think the more distributed the training is, the more likely it is to be relevant for your future, and you should remember it. So I think that's why we've evolved memory processes that are more effective when stimuli happen in a distributive manner, and it allows us to not overrepresent events that happened really close together and likely only has to do with that particular circumstance, um, and not have broader implications on your life.
Mm-hmm. Mm-hmm. For sure. Yeah, I think when I was first learning English, actually, if I, for example, if you remember the words and memorize the words for that moment, if you
Mm.
don't use the words again, you basically just forgot it. So basically, repeating
Yep,
and distributed training, using the words and again, it helps you to memorize it even more and it makes it more natural, right?
absolutely.
Mm-hmm.
Absolutely.
Okay,
Oh, and
awesome.
also it mimics distributed training mimics you actually needing the words. So like for
Mm-hmm.
example, you might use the words because you need them or because you're training them, but on a neurological level, like an individual neuron level, there might not be that much of a difference. So you're, activating the same, for example, pathway of neurons.
Mm-hmm.
Maybe you're doing it for training, but your brain is like, oh, this came in useful. Maybe we should keep that. And so that's another reason that you could have evolved. We might have evolved to remember things that occur further apart in time.
Mm-hmm. Okay, awesome. So that's about your research project in the future. So how about your own, for example, in the future, this year or next year, any bucket list that you want to do in the future for now? What do you think?
Like career-wise,
It can be career-wise or
or
just
just like
something
a
like the
research, a
travel you
research
want to do in
project
the future.
or
Yeah.
travel.
Yeah, anything.
Like, well, we are planning on getting calcium imaging working in the lab.
Wow.
So calcium imaging is where you can so calcium is a ion that cells and neurons um, often use to uh, initiate internal processes or modify internal processes. It's a really important signaling molecule. And in neurons, especially in C. elegans, in humans as well, when calcium enters a neuron, it's usually a trigger for activating the neuron or
Mm-hmm.
activating downstream processes. And in C. elegans, they don't have voltage-gated sodium channels like humans do. So calcium is actually the ion that's depolarizing the neuron. So if you can image intracellular calcium levels, you can essentially image when the neurons are activating and deactivating. So we're trying to get that up and running. To do that, we need to build a microchip or a number of microchips that the microfluidic chips that you can put worms in, and it will, through fluid pressure, hold them still so you can image the calcium in the neurons.
Mm-hmm.
So to do that, we're gonna have to build it. To save money, I'm gonna be trying to actually build it.
Oh,
I do
wow.
not have an engineering background, so
Mm-hmm.
it's going to be interesting. But I've done the first steps towards it. There's a room at UBC called the clean room, and
Okay.
it's essentially the air is extremely pure, and there's very, very, very like I think like 100,000 or a million orders of magnitude cleaner than the average air. So there's very few particulates in the air. And so that lets you use equipment that's very sensitive. And so you actually, it's called lithography, where you use light, so UV light, to actually change the properties of a mold, and so you can use this light as like a laser to, this is my understanding of it, to make these minor alterations, tiny alterations to these chips, which then I can use for the microfluidics.
Mm-hmm.
So yeah, I've been working on that, and it was really cool. I got to go in there, and there's like several rooms when you're entering of different, each one's higher and higher pressure until you get to the clean room, so that the air is always blowing out. So all the
Wow.
so and then you do like an air shower as well, and they've got like not a hazmat suit, but it kind of looks like a
Mm-hmm.
hazmat suit on. It's quite fun.
Wow, for sure. It's like sounds like the one in the movies, right? When it get into these very
Exactly,
big
exactly.
laboratories, there is like the all the mist is coming out, and then you're
Yep.
going inside with this very like the important clothes, right?
Yeah, yeah. So exactly. So that was a lot of fun.
Wow, yeah, that sounds like a fun. Awesome. That sounds like a very exciting project. Hope you're gonna succeed in
I
that
hope so
project.
too. It'll
Yeah.
add a lot of really important data to my project. So fingers
Yeah, for
crossed.
sure. Okay, so I think lastly, I think I would like you to how look back into your academia career and to get to this position. How hard was it for you? And from looking back and learned experiences, what advices would you give somebody who is trying to aim for this academia and going for this PhD in research, and
Yep.
specifically even for the neuroscience? And give us advice to listeners and for me, everybody who
Yeah,
is out there
absolutely.
trying.
Absolutely. I love helping people because I had a really hard time, not just figuring out how to do this because I don't
Mm-hmm.
have anyone in my family who has gone into academia, and I
Mm-hmm.
didn't have any friends who were doing it, and so it was really intimidating. Like, how do you do it? What's the life even like? Yeah, I had no idea. So I kind of mentioned some of the tips, like, but I'll start from the beginning. So it
Mm-hmm.
really helps if you are physically able to spend time in people's labs that you are interested in. So volunteering in labs while an undergrad is really beneficial and
Mm-hmm.
can give you relationships that either can directly let you join that lab as a graduate student, or maybe they can recommend other places you can go. Um, so that's like almost that's an easy way to get in. If you establish a relationship by volunteering in someone's lab and do a good job on the work, they're much more likely to let you into the lab as a graduate student. Again, they might not have the money, but if they do, you're much more likely to get in. And professors have a large influence on who gets into the program. So if your marks aren't high enough, but a professor really wants you in their lab, they can make that happen.
Mm-hmm.
Um, so that's something to keep in mind. Now it's harder if you're not in the city or the country that the where you're applying. Obviously, you can't do that, so that becomes harder. Um, so in that case, when you email them, make sure to make it obvious that you've spent an effort to know about their research specifically. So don't
Mm-hmm.
just email spam 1,000 professors at the same email and you just change the names. So what I would do is I would spend a day looking at a professor's papers. So I read several of their papers, think about why I'm interested in their work, and then I would include that in the email. I still wouldn't really know that much about their work. Like my understanding level of their work is still fairly surface level at that
Mm-hmm.
point. But it's enough that I can add a paragraph or two to my email about why I'm interested in their work. And it's clear I've put an effort and I'm not just copying and pasting the same email over and over again. Because professors get those kind of emails every day and they just ignore them because they get it
Mm-hmm.
every day. Um, and so that's a really good way. Of course, if you know the particular professor you're interested in, then you can make an even, you can go crazy on the email. But if you have like you're like I want to go to this university and I'm interested in a number of these labs, that's the uh, approach I would recommend.
Mm-hmm.
And I wouldn't recommend getting too attached to a particular lab because even if it's a perfect fit, they just might not have funding for you. And so
Mm-hmm.
you've got to be realistic and not just put all your eggs in one basket. Or you could
Mm-hmm.
wait a year or two and then they'd have funding, but Yeah, I wouldn't recommend that. Um, so yeah, so that's some practical tips. Um, yeah, and so I'd say also once I did that and did the work to like actually contact these professors,
Mm-hmm.
that was the like hardest part was actually getting myself like to figure out, okay, this is what I need to do to contact them and to this is how I can get success by contacting them. But once I did that, it wasn't too hard. Like I got a lot of no's or don't have funding, but I got some positive responses. I go to do an interview.
Mm-hmm.
They usually, at least for UBC, maybe Harvard or someplace like that's different, but they're not expecting the world of you in the interview. They're just like they mainly want to know like you're interested, you're gonna do hard work, and that's the main thing. If you to sweeten the deal, you can offer to start in their lab like early, and if you do good job, then they can, sign off on your application, for example. So that's something, if you're able to do that and you're really interested in the lab, you can definitely do that um, to prove to them that you're worth it.
Mm-hmm.
Um, because professors are taking risks with students when they agree to take them on.
Mm-hmm.
They can only, unless you're in like a massive lab, you can only take on so many graduate students. And when you get duds, it really slows down your lab and your paper production, your research production. And they need these uh, papers and achievements to get their next funding and grants.
Mm-hmm.
Um, and every professor gets some duds or graduate students who don't work out. And it's not that they're stupid or anything, it's just science might not have been the profession for them.
Mm-hmm.
Um, yeah. Let's see, anything else I can say? Well, I guess being in graduate school has actually been a great experience for me. I was really
Mm-hmm.
stressed out about it, and again, as I mentioned, I didn't know anyone who did it, so I had hard time imagining myself in graduate school and what I would do all day. But you figure it out, and you gain confidence as you do successful experiments, as you solve problems. You gain confidence, and after the first year as a graduate student, I was way more confident in my abilities.
Mm-hmm.
So that's something else I would say is don't let your anxiety defeat you before you've even tried.
Mm-hmm.
Um, yeah, because I'm an anxious person and definitely stressed about it a lot. But once I got it there, after a year, I was much better place.
Mm-hmm. Awesome, awesome. Those are very valuable and great advices.
I hope so. Yeah.
Yeah. And thank you, Nick, for joining today. It was a lot of great topics and a lot of great explanations. And I think we had a great conversation on these different topics. And thank you for joining me today.
Absolutely. It's nice seeing you again.
Yeah, of course.
And
It's
it's
very
great
nice
talking
seeing
to
you.
you.
Yeah, of course, as we used to always talk in office hours.
Hmm.
And I really wanted to share this to the people who are out there. And then there's a lot of like many interesting topics that we talked in the class was very valuable and then people might can use in their lives. And neuroscience is not just about all the cells and all the neurons. It's all about how we can also like there's a lot of application in the real life. You know how to use it,
Absolutely.
right?
Yep.
Awesome, thank you, Nick.
All right.
So probably after you're finishing those big projects and hopefully I'm gonna bring you back here and talk about those projects.
Sure.
Yeah, awesome.
Absolutely.
Thanks, Nick. See
All
you
right.
later.
Bye.